Hello, and thank you for listening to my poster. I will talk about understanding the trajectory of differentiating monocytes in the inflammation process of atherosclerosis and in a LATAM healthy population. This project was developed under the Project Jaguar and in the Instituto Universitario de Ciencias Biomédicas de Córdoba in the lab of Dr. Danilo Sestin. Why this is important? Cardiovascular diseases are the leading cause of death worldwide and often result from advanced atherosclerosis, a chronic inflammation condition. Monocytes are essential in the initiation and progression of the atheroma plaques due to their ability to adhere to vascular walls, transform into macrophages, and accumulate lipids, ultimately leading to the formation of the atherosclerotic plaques. During this process, different chemokines, adhesion molecules, and receptors such as CD36 and low-density lipoproteins receptors are involved. The expression levels of these receptors change through the different stages of monocyte differentiation, impacting their roles in the endocytosis of modified LDLD, a crucial factor in the disease progression. Given the importance of the receptor as a modulator of the key signaling pathway in the immune and inflammatory responses, this study focused on characterizing the expression levels of CD36, LRP1, and other factors in monocytes, as well as studying the differentiation trajectory into macrophages and in the development of the atherosclerosis. How we are going to do this? We are going to study a cohort, an Argentinian cohort of Jaguar, where we did single-cell RNA-seq. We also are going to compare the results with databases of bone marrow or coronary artery of patients that has already the disease. And basically, this is the process of how we collect samples for Jaguar. They do a survey, we process the PBMCs, then we are going to do the selection of the pipeline that we develop, also do marker exploration and try to understand the trajectory analysis of monocytes. We are going to study, like I said, in Jaguar patients and also in additional GEO databases. The general workflow is a combination of SERAT and Monocle 3, where we pre-process the data and normalizing, removing batch effects. Then we do the nonlinear dimensional reduction, clusterless cells, and compare the clusters before doing the trajectory analysis. Like I said, we are going to study Jaguar patients in PBMCs, also other GOE databases that has chronic diseases such as diabetes, for example, or obesity. We want to understand what's happening with the monocytes, with the different monocytes and macrophages. Our preliminary results. In order to put the pipeline ready to work, we start using this data set of already published project where we selected monocyte as I'm showing here. We were able to identify 8 different clusters and through annotation identify only 6 of them. But we were able to do trajectory inference and also pseudo-time inference of these populations. Also, we are able to identify canonical markers for monocytes and for macrophage. We are very interested in knowing, for example, CD14 is one of the characteristics, one of the populations of monocyte markers. Also, LRP1 is one of the markers that we study here in the lab. We are able to identify in those populations. Also, we were able to do a pseudobulk gene expression where we can see the different genes that characterize these specific populations. The preliminary results for PBMCs, we still don't have our data to analyze it, but we are able to run through the pipeline. We can see the clusters here. Also, we try to do different annotations to see which one can identify better these monocyte populations. We're able to do, for example, this dot plot. We can see all the markers they are expressing in each one of these clusters here. We see that mainly clusters 0 and 9 are CD14, 0 and 9 are CD14, giving us idea that these clusters here can be monocytes. When we do the manual in-house annotation, we can see that differentiate CD14 monocytes and CD16 monocytes. We get different results when we do the annotation with a azimuth. For example, level 2, we can see CD16 and CD14, platelets and dendritic cells. When we use a reference, for example, with single AL, the immune cell expression data as a reference only can differentiate two populations of monocytes. If we use a reference, a bone marrow can identify some of these cells as stromal or like undifferentiated type of cells. And if we use, for example, with single R blueprint and code data, only has one classification for monocytes. So we decide to stick with this one. That was the broader classification, and try to do trajectory analysis with this classification of monocytes. Also, we're able to see the canonical markers, CD14 and lysozymes, and CD16 and MS4A7 that are characteristic for the other population, the no classic monocytes. We can see they are present in our clusters. But when we try to do trajectory, we don't see a clear trajectory in these populations. We see kind of three big different clusters, meaning that we need to work further to identify if these clusters are really monocytes or are other population. Or the other hypothesis that we have is that this cluster don't differentiate into them. If you know, there are three different populations that comport and have different characteristics from themselves. So in conclusion, we successfully developed a robust pipeline for trajectory analysis. Our preliminary result in bone marrow shows 7 different clusters, but we are able to annotate only 6. We identified several genes, including LRP1 and CD13, and whose expression levels change during the differentiation process from immature monocyte to macrophages. In PBMC, we successfully identified clusters associated with monocyte, but we encountered difficulties, like I told you, annotating these different subtype of monocytes. We should investigate this issue further. And the next step is to analyze the Jaguar data and also compare it with other chronic diseases that has alteration in macrophages. And finally, we want to acknowledge the people that support this work, the CCI, the Jaguar Project, and the CONICET in Argentina. Thank you very much for your attention.